We all see beauty in the plants and the flowers that we see growing in our garden. But there's actually even more beauty hidden deep inside plants, a beauty that's at the microscopic level. Well, I'm Dr. Phil Hands. I'm an experimental scientist in Canberra at CSIRO, which is Australia's national science agency. And we do a whole range of research on food and crops that are important to Australia. I lead the Black Mountain Microimaging Centre, or BMIC for short. Quite a specialised microscopy facility in Australia if we're looking at plant samples. Using microscopes in a way that allows us to see the tiniest things that we can't see with the naked eye. So what's amazing and very often surprising as we're imaging is that even though what we're doing is producing the data to answer very specific research questions, in many cases what you're actually seeing is something that has perhaps never been seen before. It's a thing that we would absolutely never ever be able to capture unless we were harnessing the power of microscopy. And it's often really beautiful. So quite a few of the images that have been produced over the years have actually won awards. So this is one of our amazing images, won a pretty prestigious award actually, the Eureka Prize. So what you're seeing here is actually the flower in its very earliest days, the flower of a wheat. So right when that wheat plant's quite small and the flower's tucked right down inside it, perhaps just about level with the ground. So I grew up in the UK, in the Midlands, close to Birmingham. Throughout my childhood, I suppose, I've always been fond of being outdoors. Trees were always my thing, being in the forest. But I ended up doing a plant science degree at the University of Worcester, and I loved it. And ultimately, I came out of it with a PhD. And so I started to look for a postdoctoral research project. And just before I did that degree, I actually came on holiday to Australia. But within a few weeks of being here, I pretty much decided, myself and my partner, this was a place we really, really wanted to live. And in 2013, I moved from the UK over here. So I came to work at CSIRO in Canberra. It's a bit of a dream job. The typical way it works in BMIC is that someone will come to me with their sample and what they want to see in their sample. And it's up to me to try and help them find a way to use microscopy to actually see what they want to see at the time that they want to see it. I've got some samples in the critical point dryer. So I think if you can get those out, and we'll get them gold coated and get them down onto the electron microscope. So very often in our research, we're looking at things which are on the surface of plants and their organs. When we do that, the machine that we use is our electron microscope, because it has such a fantastic range of magnification. It's quite different to microscopes where we're using light and glass lenses. So you've got a stream of electrons hitting your sample and bouncing back up to our sensor, and from this we can make these amazing high-resolution images. This is actually the surface of a cotton ovule, what's going to grow up to be the seed. It's about 1 millimetre long there. So the magnification there is about 50 times. But with this machine, we can go up to 5,000 times magnification. So cotton fibre actually comes from the seed, and on the day the flower opens, some of the cells differentiate as cotton fibres. So an individual cell starts to grow out from the surface of the seed. It's actually the longest single cell in the plant kingdom. The other thing you can see in this image, actually, on the surface of the ovule, some stomates. So these are these little structures that allow plants to breathe. They're shaped like a mouth-shaped structure. What you're seeing here is actually a trichome, the hairs that you get on the surface of the leaf. So trichomes form like protective barriers against pests. Trichomes also do things like protect leaves from sunlight. We have a couple of fluorescence microscopes using fluorescent light. Laser scanning confocal microscope is really our flagship, specifically for working on plants. Our microscope has a really powerful laser. If it wasn't so closely guarded, you can actually burn holes in the wall. That's how much light we want to get through to our samples. We can really zoom in and take just a particular part of the light and shine it on. So when you're looking at me, you're seeing white light, so that's everything from blue up to sort of like red light. With fluorescence microscopes, we're using a much more specific part of that light. And we're going to shine a specific light onto a dye or a stain. What they do for us is then help us to physically see things which we can't actually see. This is one of our award-winning images collected on our laser scanning confocal microscope. It's actually the surface of a cotton leaf. And images you collect on the confocal microscope, actually, they're all collected in black and white. And we add a false colour in at the end. And you can see these chloroplasts, these little structures in the cells. And we've coloured that blue. And then in pink is the surface of the cotton leaf. Often it's not actually the most beautiful image that is the most meaningful or the most valuable. And actually the vast majority of images that we capture, they're not truly beautiful images, but amongst them, there can be the thing which tells you the most. So what you can see here is actually the ovule of a little plant called Horaceum. So that big oval structure is actually the whole ovule. And then that thing that looks like an eye, that's actually the egg cell. So that's what's going to grow up to be the embryo of the plant. And just to the right, you can see a line, some horizontal bars. So that's the DNA inside that nuclei, all lined up into its chromosomes and splitting apart. And so what we've actually captured there is the very first division of the endosperm, that critical part of the plant that's going to form all the nutrients to feed that seed. And I looked at very many images like this, and I was only able to capture this 1 or 2 times. So you see wonderful things in even the most obscure samples, but as a consequence, you've got to start to make choices because you can't look at them all. So I have to choose the projects that are most interesting and are most valuable.